All right, I think we'll get started. Hi, everybody. My name is Bradley Cooke, and it's a real pleasure to be able to share this with you. I see some familiar friends and faces. It's great to have you all here. I have been a professional neuroscientist for 25 years now, and so it's my great pleasure to be able to share this neuroscientific theory with you about how psychedelic drugs like magic mushrooms, LSD, and MDMA affect the brain and consciousness. The advent of the psychedelic renaissance has been a boon for neuroscience because these substances are not only a tool for awakening and personal growth, they're also an amazing tool for science because they have taught us and are teaching us a lot about how the brain works and about the origins of consciousness. So that is the audacious goal of this talk, is to share with you what we've learned about human consciousness from the use of psychedelic drugs. So in this talk, I'm going to be offering a theory about how the brain works and produces consciousness. And this is not my own work, it's not my own theory, but it's a synthesis of many other people's ideas, including Carl Friston and Robin Carhart Harris. So I look forward to, if you have any questions during the talk, need clarification, please raise your hand. I love a discussion and interaction. Welcome, please come in and sit down. So to begin this talk, I want to share with you a scientific instrument that was developed at Johns Hopkins University called the Mystical Experience Checklist. And this is just a sample of it. It's actually much longer than this. But these are a few items on the Mystical Experience Checklist that, if a subject endorses any number of these, then there's a good bit that that subject was having a psychedelic experience. So let me just read some of them to you to give you a sense of what this checklist is all about. So if a subject endorses the following things, again, we might think that they were tripping. So there could be a sense of reverence, an awareness of the life or living presence of all things, a feeling that you experience something profoundly sacred and holy, the experience of the oneness or unity with objects and or persons, a feeling that you experienced eternity or infinity, the experience of unity with ultimate reality, and finally, the experience of pure being and pure awareness beyond the world of sense impressions. So hopefully this conveys some sense to those of you who have not taken psychedelics, what it's like to take a drug like LSD or psilocybin. Now, this being a science-based talk, I love data. So the next thing I'm going to show you is some data from a paper published in Psychopharmacology. And in this study, 30 adults received psilocybin, the active ingredient in magic mushrooms, and a control substance, which in this case was methylphenidate, or as we know it, Ritalin. And they received these drugs on different occasions and had an experience elicited by the drug. Afterward, they were given a questionnaire. And one of the questions was, how personally meaningful was the experience to you? And the bars indicate the percentage of volunteers that endorsed the following statements on the y-axis, or listed here. So if you look, the black bars represent those subjects that received psilocybin, and the hatched bars represent those that received Ritalin. You can see that of those that received psilocybin, they considered that to be among the top 5 most personally meaningful experiences in their entire life. And when those subjects were asked, what are some other personally meaningful experiences? What are some others in the top 5? Subjects would say things like witnessing the birth of my children, or the death of a parent, things like that. So to me, this kind of data indicates that something profoundly meaningful and important is going on with psychedelic drugs, and it behooves us to understand how they work in the brain. So for now, I want to talk about how psychedelic drugs have the effects that they do by providing you with a quick sort of Cooke's tour of the brain and how it works. And from there, we'll delve into the effects of psychedelic drugs on the brain. So to begin, I'm gonna try to provide a sort of basic level picture of what we call conscious perception. How do we form pictures or images in our brain when we sense something? And to begin this story, I want you to consider the fact that inside my head and inside each one of yours, it's dark in there. There is no light inside of our heads, and yet the brain produces light somehow. How does the light get in there? Well, the short answer is that the brain generates it, okay? The brain generates perception. And that's going to be the sort of underlying theme of the next few slides. Okay, so here is a schematic of the human brain as seen from the right side. This is the cerebral hemisphere. And the first thing to notice about this schematic is that there's a wrinkled surface on the brain. I'm sure this is a familiar picture to most of you. That wrinkled surface is called the cerebral cortex. And it covers the entire surface of the cerebrum, as we call it. The second thing to notice about this is that localization or location equals function in the brain. And this schematic illustrates that. So, for example, at the back of the brain, shown in green, is the visual part of the brain, or the visual system. In dark green is the primary visual cortex, and light green is the visual association area. And if you continue looking at this picture, you'll see that there are different areas for each of the senses. So, for example, in the temporal lobe, there is the auditory cortex. This is where the conscious perception of sound occurs. And next to it, adjacent, is the auditory association area. In blue, at the top of the brain, in the parietal lobe, is the primary somatosensory cortex. This is where the sensation of touch is represented in the brain. And so on down the line. Every one of our senses of the external world has a map or an area in the brain that's dedicated to it. Olfaction, taste, hearing, and so forth. Each one of our senses has a distinct area that's dedicated to that function. Okay, now if we were to take a coronal slice through the brain and look at it, this is sort of what it would look like. You can see the wrinkled surface, the cerebral cortex, around the perimeter. This is the white matter, and the cerebral cortex itself is gray. Now, if we had an imaginary microscope and we could look and peer at the cerebral cortex itself, excuse me, we would see that the cerebral cortex has 6 layers of cells. And the artist has illustrated what these layers look like. So here's layer 1 through layer 6. The dark images or dark things here in this picture are the cells that constitute the cerebral cortex. Those cells are called neurons, and they are going to be the subject of the next several slides. Welcome, please sit down. Thank you. Okay, in this picture, we have enlarged the picture of the cells of the brain to illustrate the neurons and some of their interrelationships. So we have a large pink cell that's called a pyramidal cell, and then there's various names of these other cells, okay? So, but the key takeaway here is that neurons have specialized structures that are called dendrites. These dendrites are like beautiful, magical trees that receive information from other cells. If you direct your attention to the large basket cell in black, you'll see that it has the thick dendrites that emerge from the cell body. And then there's a thin and very long process that terminates in large sort of little, they're called boutons. These boutons are a way in which the basket cell communicates with the pyramidal cell. And the context that we're discussing here are called synapses. Okay, so the takeaway from this slide is that neurons influence each other through synapses. The second thing I want to tell you is that neurons communicate with each other through a chemical language that is defined or classified by the term neurotransmitters. So neurons produce neurotransmitters and influence each other through these synapses using neurotransmitters. If we were to take our mental microscope and enlarge this picture even further, here is a cartoon of a synapse. In blue is what we call the presynaptic cell, and in pink is the postsynaptic cell. Information in the nervous system travels in one direction. It goes from the presynaptic to the postsynaptic. And now I want to explain to you very briefly just how that works, okay? So the language of the brain is not only chemical, it's also electrical. So neurons fire something called action potentials. These are tiny little impulses of electricity that travel down the axon and reach the end of the axon, called the axon terminal. When that little pulse of electricity reaches the axon terminal, it induces the release of neurotransmitters into the synaptic cleft. The neurotransmitters that enter the synaptic cleft diffuse randomly and may come in contact with a receptor on the postsynaptic cell. When this happens, the receptor induces an electrical change in the postsynaptic cell. And that could make that neuron more likely to fire an action potential itself. So the language or the process that we're discussing here goes from electrical to chemical to electrical, all right? Now, one of the neurotransmitters in our brain that's very important for this story is serotonin. Okay, you've probably heard of serotonin. It's an ancient molecule. It's found in every creature with the nervous system, uses and produces serotonin. And just like any other neurotransmitter, serotonin works in this way. When it's released into the synaptic cleft, it binds to receptors and causes a change in the excitability of the postsynaptic cell. And of course, serotonin is at the heart of the story about how psychedelics work in the brain. So we'll be returning to the story of serotonin in a few minutes. The next thing I want to tell you is we're going to actually use our mental microscope and zoom out a little bit now and talk about another aspect of organization of the cerebral cortex. So in this picture, we have the cellular constituents of the cerebral cortex and their connections. If we take a step back a little bit and look at the cerebral cortex from a greater distance, we see that the cerebral cortex is composed of columns of neurons. Okay? These columns of neurons comprise approximately 142 cells, and they tile the cerebral cortex. The entire cerebral cortex, from top to bottom, left to right, is tiled by these microscopic columns that extend from the top to the bottom. What's interesting about the columns is that the connections amongst the cells within each column is kind of stereotyped. It's a common kind of connectivity from column to column, no matter where you look. Okay? The second thing to know is that these columns are kind of like the brain's little computer. They take the synaptic input from another column, transform it in some way, and spit that information out, sending it to another column. So just like a computer algorithm, they're in the business of transforming information from one state to another. So again, we have the cerebral cortex, where different senses are represented in different areas. We have 6 layers in the cerebral cortex, and then we have these columns that go across the surface, or go from top to bottom. Okay, continuing on with the neuroscience a little bit further, the next thing I want to tell you is zooming out again. We're now thinking about the brain as a whole and our senses within the brain. Yes, question? Right, the question was, are the columns everywhere in the brain, and are they stacked on top of each other? Was that it? Yeah, they're next to each other, exactly. So the cerebral cortex is about 1 millimeter thick, and the columns are aligned with each other next to one another. So they go from layer 1 to layer 6 everywhere in the brain. Okay, and then again, there's connections within and connections between those columns that neuroanatomists have a lot of fun trying to figure out. Okay, yes, another question. I'm not sure, can you repeat that?
So with the cells themselves, are the cells themselves all oriented the same, or do they change each part of the brain?
Oh, that's an interesting question. The orientation of the cells is quite stereotyped in the cerebral cortex, right? So they often have a dendrite that points upward toward the top and other dendrites that point toward the bottom. It's quite, yeah, there is some anatomical regularity in terms of the way they're oriented. What I'm speaking of now, though, is true about the cerebral cortex. There are many other parts of the brain that we're not going to discuss today, in which those ideas are not necessarily true. Okay, so now, if I may, go on to this next idea, this next concept, which is that each sensory area in our brain is a map of its surface on the body. Okay, so consider, for example, the retina. Okay, the retina is the sheet of tissue at the back of the eye that, if it could be dissected, could be laid flat on a table like a napkin. Okay, it's a two-dimensional sheet that lays at the back of the eye. And we can define an XY coordinate on this sheet of tissue. And in the very same way that we can define it on the retina, so too is there a particular and specific region in the visual cortex that responds to light at that position. Okay? Take another example, the skin. The skin is another surface for which there is a map in the brain. If I were to stimulate my middle finger on my right hand, there's a unique and particular region of the cerebral cortex that responds to stimulation in that area. Okay, this principle of mapping is true of all sensory systems. And so it's a ubiquitous common feature of the nervous system. So we have maps in the visual cortex, auditory cortex, somatosensory cortex, and so forth. Now, just as there are maps of the external sensory environment, we have maps, again, for the visual system, the somatosensory system, auditory system, so too are there maps for the internal part of our body. There are maps that represent, for example, the inflation of our lungs, or the distension of our bladder, or how much glucose there is in our blood. Each one of these different variables is represented by a unique map in the brain that has a unique location, okay? So these together, these maps contribute to the sense of our having a body, of our having a self that persists through time and has different states. Sometimes we're hungry, sometimes we're full, sometimes we need to pee, sometimes we don't. These different states are represented in different maps, okay? Now, I just alluded to something that's important, which is that these maps may contribute to the sense of embodiment, having a body that is particularly ours, and one that persists through time and has changes that occur to it. So this is an important clue to consciousness. I would argue that consciousness is deeply involved in having a body, in the sense of having a body. So if we can look to where those maps are, perhaps we can learn something about consciousness. Recently, neuroscientists have identified a set of areas called a neural network that seems to correspond to the representation of the body. And this set of neural networks, or neural network, is called the default mode network. The default mode network seems to be involved in integrating the sensations of our body with the sensations of the outside world. Okay, so this is a master integrator of the external world with the internal world. And one of the most important and sort of salient clues from the neuroscientific study of psychedelics is that when people take psychedelics, they often experience a sense of depersonalization or ego loss. Okay, I'm not sure how many of you have had that experience, but it's a quite reliable effect. In fact, the writer Michael Pollan, in his book, How to Change Your Mind, described depersonalization as a sense that his ego had become so flattened that it was like a paint that covered the surface of every nook and cranny in his room. So depersonalization is a common feature, and we believe that the default mode network is involved in the sense of having a self. So it's not surprising then that correlations have been discovered between being on a psychedelic and the and influences on the default mode network. Again, one of the most reliable findings from the neuroscience of psychedelics is that the deactivation of the default mode network is a very common feature. This is a functional MRI scan of the brain, showing it from various perspectives, and the blue colors represent a deactivation of the brain. And the one region of the brain that is common to all of these pictures is the default mode network. So this seems to be a brain behavior correlation that psychedelics has revealed to us. Are there any questions at this point? Yes, sir. Well, the psychedelics I'm referring to in this talk are those that activate the serotonin 2A receptor. So this includes the phenethylamine psychedelics and the tryptamine-based psychedelics. Although depersonalization is a common feature for many others as well, including ketamine, for example. Okay, so I'm going to now step away from the neuroscience and get a little bit more theoretical, because our goal here in this first half of the talk is to talk about conscious perception. How do the images get inside of our brains? And to do this, I'm going to offer some metaphors, some ideas that hopefully help you understand where I'm coming from, although they're not meant to be taken literally. So in the history of psychology and neuroscience, there have been a lot of ideas about perception. And one of the most common and persistent is the idea that perception is an outside-in kind of process. And I've tried to illustrate that with this schematic. So here's the eye, here's the optic nerve, and this staircase is meant to represent the brain. So the concept is that images fall on the retina and they're transmitted to the brain, and then one region of the brain recognizes simple features, and that passes the information on to another region which recognizes objects, and yet a third region which recognizes and forms concepts, and yet another region that's involved in metacognition. Now this theory, or this idea, while very intuitive, has a major problem with it, a kind of philosophical problem, which is that it leads to an infinite regression. Namely that if this is where the images are formed, who then is viewing those images? Who then is responsible for actually seeing the objects, concepts, and manipulating ideas that this theory would purport they do? That this concept or this problem is illustrated with a cartoon like this one, right? So here's the brain, here's the eye, and here is a little ghost inside of the machine viewing the images displayed on the cerebral cortex and making decisions about what to do, like to move or to think or things like that. This is obviously not correct. There is no ghost in the machine. There is just the brain. So we need a better theory that explains conscious perception. And what I'm going to do now is try to explain that to you. The new idea is that the brain is fundamentally involved in prediction, okay? That the brain at the cellular level and at the highest level is involved in prediction, specifically predicting its own sensory input. And I've tried to illustrate that with another metaphorical picture. So here is the familiar eye and optic nerve, and the S's represent sensory information that's rising through the brain, going from primary sensory areas to later sensory areas. And again, we have the familiar recognition of simple features, the identification of objects, concepts, and so forth. But just as important in this idea are predictions that descend from these higher levels and influence the processing of sensory information as it comes up through this hierarchy, okay? So this is called the predictive processing model of brain function, and it's quite current and sort of trendy right now in neuroscience. And again, it says that at a fundamental level, the brain is a prediction machine. At the same time, however, not only is it making predictions, the brain is attempting to, okay, this is a mouthful, to minimize the discrepancy between the prediction and the actual input that it's getting. Okay, to minimize the discrepancy between the prediction and the actual sensory information that's coming in. In fact, this theory says, the content of our consciousness is the discrepancy. It is the difference between the prediction and the actual information that comes in. And this can be defined mathematically, that the numerical difference between the prediction on the one hand and the actual on the other is the content of our consciousness. And I'll leave it to you guys to think about that on your own, but I'm just going to leave that there as we continue with this, this theory. Now, I want to emphasize that it's dark in there. It's dark in there. There's no light inside of our heads. The brain has to generate its own images. And that generation, I would suggest, looks something like this. Here is the external world, a real place that is actually out there. Our senses interpret that information and generate a model of the world. That model is generated by the brain And it consists of prediction and the sensory signals in a dynamic interplay. And again, the content of our consciousness may be that discrepancy between the prediction and the actual. Are there any questions before I move on? Okay, I'm gonna now take this idea and make it a little bit more concrete at the neurobiological level. So you'll have to bear with me if I have a little more, some cartoons that I've made. Okay, you remember the cortical columns? Those cortical columns, again, can be found throughout the brain, every sensory area. So we have primary, secondary, tertiary sensory areas. Here is a cortical column in, say, the primary auditory cortex. And here are the familiar sensory inputs that are passed from one area to the next. And at the same time, this theory says, we have predictions that descend from later areas to earlier ones. And I'm going to begin calling these predictions prior beliefs, or priors, okay? This reflects the heritage from a branch of statistics called Bayesian statistics. And so we have these priors that influence and constrain what the sensory inputs are. This process of priors influencing sensory beliefs is occurring in the brain as an oscillation, okay? A back and forth, a rhythm that occurs around 10 cycles per second. Okay, it's also known as the alpha rhythm. And again, let's see if I can, yes, so this alpha rhythm generates a prediction error, or the discrepancy between the prediction and the actual. This is just a bit of a digression, but I want to point out that this idea of brain activity occurring in an oscillation is a very common one in neuroscience. And I just want to illustrate what that means for a second. So here's the visual cortex. And if you were to put an array of electrodes on the surface of the brain, we would see that it could be cycling at approximately 40 cycles per second, or 40 hertz. And there might be other regions that are operating at the same rate. And some believe that this could represent a kind of synchronicity between areas that might be involved in the generation of consciousness, although we don't really know for sure. Now, much more importantly, though, is the idea that this use of the importance of prior beliefs on our perception can give rise to interesting illusions that we now sort of have an explanation for. So who can see the white triangle in this picture? Raise your hand if you can see the white. Good, right? So if you were on a psychedelic drug right now, you could not see that, right? This is called the Kniza triangle, very common in introductory psychology textbooks. And we think that the appearance of the white triangle represents the brain's best guess as to what this sensory information could represent. The brain seems to think that rather than these being little Pac-Men, instead these are actually black circles that are obscured by something in the foreground. This is a prior belief that pertains to like what's in the foreground obscuring things in the background. Okay, that's a prior, maybe developed through evolution, that helps us interpret the visual world. There are other examples of this that are interpreted in the same way. So here's a sort of a Loch Ness monster-like figure, right? Most of us sort of infer or guess that there could be something beneath the surface of the water leading to this undulating pattern. But in fact, there isn't. It's just 3 independent images. It's our brain's inference about what's there that makes us think that this could be some kind of snaky creature on the surface of the water. Perhaps the most famous example of perceptual priors influencing our consciousness is this next image from psychology textbooks. This is the famous duck rabbit, okay? So as you view this, your consciousness may flip from perceiving the image as a duck and perceiving it as a rabbit. And I'm sure thousands of psychology students have been asked to press a button whenever they perceive that flipping to occur. And we think that this represents the application of 2 priors to the image. One prior being that these features represent a duck, the other prior being that these features represent a rabbit. Interestingly, very few people seem to be able to perceive a duck rabbit simultaneously. It can only be one or the other, implying again that there's a kind of oscillation between priors. Okay, so I've given you a whirlwind tour of the way the brain may work, and what I want to do now is introduce the subject of psychedelics. And we're primed to begin to appreciate this because researchers in this field have suggested that what psychedelics may be doing is relaxing our prior beliefs, okay? That the priors under psychedelics are no longer so strict about what comes in. Instead, they have been relaxed. And so, in contrast to perception at baseline, where there's a healthy balance between the information coming from our senses and the influence of prediction, if predictions have been lessened, then the influence of sensory inputs becomes significantly greater. So, to carry this one step further, if the effect of priors is relaxed, then the influence of sensory input becomes that much greater. And it can actually lead to the brain becoming much less constrained about the way it works. And researchers have observed that under psychedelics, brains often display higher levels of chaos and disorder, again, perhaps reflecting the relaxation of sensory priors. Okay, we're going to delve now back into a little bit more neurobiology about the way psychedelics affect the nervous system. So here is our familiar schematic diagram of the cerebral cortex, just a few of the cells. Here is a pyramidal cell in pink, and these are the beautiful mystical trees of the mind called dendrites. On the surface of a dendrite are receptors for neurotransmitters. And the receptors that we're discussing now is the serotonin 2A receptor. There's something like 9 or 10 different serotonin subtypes. So we're very lucky that the serotonin 2A receptor exists because it is the one that can bind drugs like ergotamine, which is an analog of LSD. It can also bind psilocybin or psilocin, the active ingredient in magic mushrooms. And whether it's serotonin itself or a psychedelic drug, the effect on the neuron is to make it more excitable, more prone to fire action potentials, all else being equal. So what could be the effect of this increased excitability on the oscillation between sensory input and our priors? Well, here is the familiar diagram again, but this time I'm trying to emphasize that we have activation of our 2A receptors under the influence of psychedelics. And as a result, our influence of prior beliefs is much, much lower. At the same time, prediction error goes way up. And what is it that these theorists say is the content of our consciousness? It's the prediction error. So when we talk about psychedelics expanding consciousness, what that might mean actually is that the prediction error is expanding or increasing. Okay, so I'm going to become even more metaphorical now and ask you to think about the neural network of the brain in a very abstract way. Neural networks are one of my favorite subjects, and we can talk about the way neural networks work in a kind of geometrical sense. So we can think of the mind-brain now as a landscape, a space of states in which the brain can be in, a space of states in which the brain can be in. And in this space of states, we have basins and we have hills. The basins and hills are incomplete. There's one more element, which is imagine an imaginary marble. Okay, that imaginary marble rolling around on the landscape is the brain itself, the state that it is in. And so these basins represent an attractor for the brain state. The deeper the basin, the more likely the brain is to be in that state. Conversely, the more shallow the basin, the more likely it is that the brain state will change and move to something else. Does that make sense? Any ambiguity about that? So, yes, sir. Much more generic than even that, right? So I'm talking about both emotional states, being happy, sad, and so forth, as well as states like being hungry or being tired or sleeping or being unconscious, right? Every one of those states, or being attentive, as most of you are right now, right? Each one of those states could be represented in this abstract representation of a neural network as a basin. Yeah, exactly, biological states, right, exactly. So if I were to administer a powerful anesthetic to someone, a propofol, for example, okay, a basin would emerge in their neural network that was very, very, very deep. And that basin would represent the brain state of unconsciousness. Make sense? Right? If I were to give caffeine to my 16-year-old, okay, the brain state would become much more excitable. And that would do something else to the basins and hills that represent her mind. Yes, so a dip or a valley is a place where the brain is more likely to be. Right, so the brain is our imaginary marble rolling around, okay? And so the basin, or what did you call it, a dip, is an attractor for the brain state. Okay, so the brain state rolls around and finds itself on the edge of a basin, falls in, may be difficult to get out. But if you look at the caption I've provided here, what do you think would happen to this landscape if the prior beliefs were relaxed? The basins would become more shallow. Mathematically speaking, there would be more entropy or chaos in the neural network. But importantly, the network would also have more freedom. Okay? Our imaginary marble has now more places it can roam to. Absolutely, absolutely, and that's exactly where I'm going, right, is that, you know, pathological brain states, ones that cause suffering for the individual, may be excessively deep brain states that are difficult to get out of, right? And through psychotherapy, good friends, lots of sleep, and maybe through the use of psychedelics, we can relax and make those basins a little bit more shallow such that new brain states can be achieved. All right, so you beat me to my punchline. No problem. So here is another representation of the same idea. So on the left is this is actually data. This is a data from several different neuroimaging experiments. But the author invented a very colorful and provocative way of representing it. So these represent brain patterns at baseline in the absence of any psychedelic drug. And this represents brain patterns on psilocybin. So this, I think, evokes very clearly the idea of a neural network that has more freedom to find new brain states. To be more specific, these lines represent interconnections between brain areas. And the thickness of the line represents the strength of each connection. So you can see that on psilocybin, there's many, many, many more connections, but they're all very weak and very narrow. Are there any questions before I move into the last part of my talk? Yes? Synapses? Right, so connections between brain areas, say between primary visual cortex and secondary visual cortex, is made by synapses. Neurons influence each other with synapses, right? And so this picture represents something happening at the level of synapses. They've become a lot more plastic, a lot more capable of finding new connections. Could be one way of thinking about it, okay? So the last bit of my talk I'm about to begin is rather speculative. I am not a psychotherapist trained as a psychiatrist, but I've read a lot of the literature and I want to offer a perspective on how psychedelic drugs like psilocybin could be so useful in improving mental health. So what are the implications for psychotherapy? Well, to introduce this topic, I want to posit that a healthy mind is like the blade held by this beautiful warrior. A healthy mind is sharp, flexible, and resilient, just like a piece of fine steel. And so what can we do to improve the flexibility, the sharpness, and resilience of our own minds? Well, I think it's clear that people with emotional trauma, their brains are unwell because they are in some way less flexible, less resilient, less capable of handling stress. It's almost as if there's a crack or a defect in the blade, in the metal of their minds. Over the last 15 years, there have been more than 10,000 publications in the most common biomedical database on the subject of psychedelics and mental health. And so there's been an explosion of interest in how these substances can improve our daily lives. One of the ways that they're being evaluated is in the treatment of post-traumatic stress disorder, which is a state of mind that can occur to nearly anybody, but it's most common in our veterans, men and women who've traveled overseas and witnessed some of the most horrific things that you can or maybe can't imagine. People with PTSD are crippled by a dysregulated emotional system. And psychedelic drugs may help make their brains a little bit sharper and more flexible. So in the next section, I'm going to talk about how that might happen. So I would like to suggest that post-traumatic stress disorder is not just a psychological phenomenon. It's actually a brain injury. Okay? It's a brain injury to those neural circuits that are involved in emotion. One of the regions of the brain that's so important for emotion was the subject of my own research as a professor, which is called the amygdala. Okay? So the amygdala is a region in the temporal lobe. It's below the cerebral cortex. And its key role is in the prediction of dangerous, frightening, scary things. Okay? The basic way that it works is that it receives sensory information from all of our senses, smell, hearing, vision, touch, and so forth, as well as our memories. It's constantly filtering sensory information as well as our memories for anything dangerous out there. When the amygdala recognizes something significant, it activates or orchestrates the expression of emotional behavior. Emotional behavior include things like facial expressions, stress hormone releases, autonomic responses. Each of these together contributes to the expression of a fearful response. But note that when we have fearful responses, that in turn leads to the production and generation of bodily feelings, right? When you're feeling afraid, you recognize those in your consciousness. And the notion here is that the expression of bodily feelings combined with particular sensory inputs can lead to new prior beliefs. And importantly, remember that prior beliefs represent the strengthening of particular synapses in the brain. Okay? And we've found, researchers have found, that the amygdala is the site of some very important synapses in terms of connecting particular emotional events to emotional responses. This is why, for example, the veteran who comes home from combat may flinch when he hears a car backfire. Okay? That car backfire is an innocuous stimulus, but his amygdala has learned that the sound of a powerful explosion represents danger. And so his amygdala, combined with the rest of his nervous system, is summoned to produce a fear response. And we need to think about ways in which we can help that emotional response be turned down a little bit. Maybe we can revise his priors in some way to make daily life outside of combat more manageable. So one of the drugs that's being investigated for the treatment of post-traumatic stress disorder is called MDMA. I'm not going to I try to say what that actually means, but it's also known as Molly or ecstasy. And in research, some of which was conducted at Emory University, research subjects, with all of whom had PTSD as the result of combat, were treated with either a placebo or MDMA over 2 sessions. And they were, of course, given these drugs by someone who recognized what they were being given. All the subjects received standard psychotherapeutic support during each session. But the subjects received either placebo or MDMA. And then a separate set of people who were unaware of what the subjects had received then evaluated these subjects for the presence of PTSD-like symptoms. And the basic story here is that after 3 sessions with MDMA, a significant fraction of veterans with PTSD could be considered to be in remission, that they had been effectively cured of their PTSD. In contrast, those that received placebo and psychotherapy showed virtually no change from one session to the next. So this, again, here is our placebo group, which received psychotherapy, which is the gold standard of treatment for PTSD. And here are those that received MDMA as well as psychotherapy. And you can see that the fraction of those that were in remission or had improved significantly was much greater. So this is a triumph, I think, of modern science and our ability to get over our hangups about psychedelics and begin exploring their use for the treatment of something as severe as PTSD. Okay, the next study I want to show you was published in another very prestigious biomedical journal called the New England Journal of Medicine. I hope you've heard of it. It's quite prestigious. In this study, they looked at 60 subjects with moderate to severe major depression. Okay, this is not bipolar disorder. This is clinical depression that has not been effectively treated in any other way. Half the subjects received 2 sessions, with psilocybin, receiving 25 milligrams 3 weeks apart, and placebo. Okay? The remaining subjects received a common and very effective antidepressant called Lexapro, and they also received placebo. But notice that they received Lexapro for 6 weeks, okay, compared with 2 sessions with psilocybin, each of which may have been 5 to 6 hours long. At the end of the 6-week trial, the change in depression was measured, and these are the numbers. So the change in depression was 8 points in those that received psilocybin and 6 points for those that received Lexapro. This was not statistically different, okay? They were statistically equivalent, but both groups were significantly different from baseline, right? So both groups showed a tremendous improvement in their depression symptoms as a result of either Lexapro or psilocybin. Sorry? There was no secondary measurement of the subjects. So in this study, they were measured after 6 weeks, and that was it. But I can tell you that the literature suggests that the effects of psilocybin can be quite persistent, right? So 2 or 3 sessions can lead to 6 weeks or more. That's right. The effects have been shown to be enduring. That's right. Yeah, thank you for mentioning that. Okay, so to conclude this section, I want to offer yet another metaphor of how psychedelics could be working. So this metaphor is derived from the science of metallurgy, that is to say, how, like the science of metals and how they work. Metals, like the sword in that woman's hand, are comprised or can be defined by the strength of the atomic connections amongst the atoms in the metal, okay? In a piece of raw iron, if you direct your attention to the left, a piece of raw iron has some connections with extraordinarily strong connections, extraordinarily strong atomic bonds amongst the atoms in the metal. And in metallurgy, people know that if you apply heat to raw iron, those atomic bonds get weaker, okay? And in fact, if you let that hot metal cool down very, 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 very slowly, the atomic bonds get very, very weak and much more uniform. They say that the bonds have become smoother as a result, and you can appreciate why. This is essentially a ragged landscape. Now the landscape is much smoother. So by analogy, the notion is that psychedelics influence the synaptic connections of the brain in the same way. This could be the mind or the brain of a person with PTSD. These connections are in his or her amygdala and elsewhere in the brain, and they represent those connections that have been formed by trauma. The erroneous recognition that a backfire represents a threat, right? What we need to do is weaken those connections. And the notion is that simply that on psychedelics, in the same way, those synaptic connections can become progressively weaker, such that over time and with adequate support and psychotherapy, during reintegration, the synaptic connections can become reweighted, such that overall the neural network is smoother. This is the idea, and that the idea is that psychedelics promote neural network annealing. So again, the concept is that in psychotherapy, psychedelics may relax our prior beliefs. And this in turn may help the subject take new perspectives on the events that led to so much trauma and trouble in their life. This may in turn reduce the prediction error between the actual sensory input and the prediction, okay? That in turn could lead to less what we call neurosis, less avoidance of ideas and concepts and events that cause pain. During the psychedelic trip, as well as perhaps later, memories become less fraught with emotion. And this allows subjects to consider and take a new perspective on the events that cause so much suffering in their daily life. And again, perhaps after the trip, and with, again, the support of friends or a skilled psychotherapist, the priors can become revised such that there's a better agreement between the sensory input that you're getting, writ large, writ broadly, and the prediction. So that's the conclusion of my talk. And to leave you, I've got some suggested readings. Like any good academic, I have to suggest some readings. So the first is LSD, My Problem Child by Dr. Albert Hofmann. He was the discoverer of the LSD molecule back in the 1940s, I think. How to Change Your Mind is becoming a classic in this field. But then there are two more that dwell on the predictive processing model of brain function. And the first is an article in the great Quantum magazine, To Be Energy Efficient, Brains Predict Their Perceptions. And Being You by the cognitive neuroscientist Anil Seth is a wonderful disquisition on the formation of the self and consciousness. So thank you very much for your attention. We have some time for questions, so. Yes. I think the short answer is that we don't know why there's variability. Researchers in any field do their best to minimize variability. It's the demon and enemy of getting statistically significant results. So I'm sure at this point, researchers are left scratching their heads as to why some respond better to psychedelics than others. But one thing in particular comes to mind that could contribute to variability, and that is the prior history of the subject with antidepressant medication, right? So antidepressant medication at the cellular level leads to changes in the expression of various receptors, including serotonin 2A receptors. So decades of use of, say, Lexapro could result in a downregulation of serotonin 2A receptors, as well as others. And that would be a response to the artificially elevated levels of serotonin that those drugs induce. And so we don't have a handle on, at the neurobiological level, of why, when, and where those changes may change. Those variations in expression level will occur. That could be one reason. There's also the underlying variation amongst all of us that contributes to our individual personality and differences. Those differences are driven by genetics and by our history. So that's a short answer to saying, or long answer to saying, I'm not sure. Oh, that's a good question. And the questioner asked, does psilocybin alter the expression level of serotonin 2A receptors? And I don't know the answer to that. Yeah, look it up. Oh my.
Really got you really curious, this is something I want to know more about.
So the questioner asked, what got me interested in psychedelics? Yeah, so I've been using psychedelics since adulthood. I may not look this way, but I'm actually old enough to have followed the Grateful Dead around the country. So I went to many Grateful Dead shows and dropped acid many, many times. And it was during an early episode that I realized that I wanted to be a biologist because I could see the beauty of nature I could see the interconnections of the natural world with our world. And I was certain at that point that I wanted to be a biopsychologist, a neuroscientist, because that to me is the perfect linkage between our biological nature and our consciousness. The highest aspects of our nature can potentially be understood through neuroscience. Yeah, thank you.
What age did you get into did you start during your biology?
When I was 18.
18?
Yeah. Someone else. Sure. Perhaps one more question.
So this predictive mechanism in the brain, it seems like it would cause the placebo effect, but does it also constitute basically our sense of object permanence? If we didn't have it, we have a sense of object permanence?
Yes, yeah, that's a great question and a good prediction from the predictive processing model. She asks and recognizes that by the age of like 6 months, infants recognize that when an object disappears behind a screen, it's still there, right? So that could represent the advent of that child's predictive processing of object permanence. It would be interesting to see whether object permanence is affected by the use of psychedelics. Okay, I thank you all again for your attention, and I think it's time for the next speaker.